Okay, next video featuring updated Hedgehog. I'm going to try and pre-record a bunch of these so I can get ahead and let y'all start to get ahead because we still have a lot left in this unit and we kind of need to get moving in it. So, last video was transcription. This video is going to be translation. Which is where we take the mRNA that we made in transcription and actually convert it to protein. Oops, wrong tab. So, our mRNA, we read the mRNA sequence in sets of 3 in a triplet, called a codon. And that 3-letter sequence is going to code for a specific amino acid. Because there are 4 possible base pairs and 3 letters long. 4 to the third is 64. So there are 64 possible codons that you can generate using those 4 letters. But we only have 20 amino acids in our 20 main amino acids in humans. So most of our amino acids are going to have more than one codon. So it's common to see 2 or 3 codons translating to the same amino acid. This provides a redundancy, which we'll talk more about, but it helps this process be less error prone. But it is not ambiguous. Every codon has a single amino acid that it codes for, or it codes for stop, but if you know the codon, you know the amino acid. There's never any ambiguity where it could be one thing or the other. You always know for sure. It's just that an amino acid might have multiple codons, but it doesn't go the other direction. And pretty much everything uses this same codon chart, amino acid sequence. We all use basically the same amino acids. We all have the same codons coding for each of those amino acids, which is a huge indicator that all life is related at some point and time. And I had to make sure I was recording this. So because everything shares this genetic code, it gives us an indication that life had a common origin. Okay, we are going to be talking about tRNA a lot in this section. tRNA, the T, I believe, stands for transfer RNA. I like to think of it as translation RNA. It helps me remember. And this is a special RNA segment, so it's kind of a functional RNA that has a specific amino acid attached to one end, and then on the other end it has what's called an anticodon, which this is just, it base pairs complementarily with the actual mRNA strand that we made from transcription. And so it'll attach at the anticodon and then deposit its amino acid. So a codon has an anticodon that goes with it. It's just its base pairing. That anticodon will always have the same amino acid attached to it, and so on. So let's talk about the table. You will have to be able to use a table like this. You're never expected to memorize it. And this one's nice, and it tells you to start on the left side, then go to the top. It won't always say that for you. But you look at a codon, and you say, what's the first letter? Let's say it's U. Then I go to the second letter. Let's say it's A. And then the third letter is whatever of the 4 options in here that I will pick. So we can see that for UA, there are 4 possible codons we can make. We can make UAU, UAC, UAA, or UAG. And UAU and UAC both code for tyrosine, and UAA and UAG both code for a stop codon. So it's not actually an amino acid, it just tells it to stop. So this is showing some of that redundancy. There are 2 codons here that code for tyrosine. If we look just one box to the left, all 4 of those code for the same amino acid. So you will have to be able to use these charts. We will practice with that today. The other big player in translation are the ribosomes. So this is an organelle in every single organism on the planet, and it is where proteins are made, and it is the site of translation. So it kind of holds those tRNA anticodons in place along with the actual mRNA strand, and is where the actual protein will get made. Prokaryotes, it's in the cytoplasm only. Eukaryotes, it's in the cytoplasm and on the rough endoplasmic reticulum. It kind of depends on what the protein's purpose is, if you remember way back to Unit 2. So, ribosomes have a few subunits. The thing that I wish I had put on here that I didn't is they also have a piece of RNA in them called ribosomal RNA that helps make up, I believe, the small subunit. Okay, let's actually get into it. Translation has 3 stages, and they have the same exact names as transcription. So the first stage is initiation, where it starts. To start in translation, the ribosomal RNA needs to bind to your mRNA, whatever gene you're trying to make, and it will specifically do this at a single point called the start codon, which is AUG. The start codon starts off every protein. So every protein starts off with a methionine, which is that amino acid that it codes for. And so the first thing that has to happen is this has to attach at the start codon site. So it attaches, and then we get the full complex. As you can see, there's 3 sites. You don't really need to worry too much about the internal mechanisms of this, but we've locked in our first amino acid into the ribosome, and we're ready to start building off of it. So stage 2, elongation, is where we start building off of it. What's going to happen now is that it's going to read the next codon, so the next 3 base pairs in the sequence, and the next tRNA with its amino acid is going to come in and bind at that site. When it binds, it's going to use a little bit of energy in GTP and attach it to the first amino acid. Then everything kind of moves around. All those tRNAs kind of shift position. They move one spot to the left, and then the initial one gets kicked out, and the new one that just came in picks up the polypeptide chain, the protein chain, and it's ready to accept another. So this just keeps going. We just keep reading the next codon. tRNA comes in. We transfer its amino acid to the peptide chain. Everything shifts left. The old tRNA leaves. It's going to go get a new amino acid, and we can repeat the process. This is going in the 5' to 3' direction, but it's not for the same reasons. There's no polymerase here adding stuff in. We're just making peptide bonds. But we still should have a direction to it. But it's not for the same logistical reasons that we have this direction. So we're just elongating the chain. tRNAs come in, drop off an amino acid. Everyone shifts. Old one comes out. New one takes over the chain. We keep building. Then we get to the termination stage, which is when we hit a stop codon. So that's either UAG, UAA, or UGA. You don't need to have any of the codons memorized. It can just be helpful sometimes. But we hit a stop codon, and that triggers, instead of an amino acid being added, it just caps it off with a water molecule and then breaks apart and releases the full polypeptide chain. And we have then successfully translated a protein. Some notes about prokaryotic versus eukaryotic cells with this transcription translation. We will do more on translation in class. If there's anything that didn't make sense, please write it down and ask me. But the differences between prokaryotic and eukaryotic transcription and translation. So eukaryotes have a nucleus, and this physically separates transcription and translation from happening. Transcription happens in the nucleus. Translation happens out of the nucleus, usually in the cytoplasm or on the rough endoplasmic reticulum. So they're physically separated processes. Prokaryotes do not have a true nucleus. They have a region where their genetic info is, but there's no boundaries or barriers. And because of this, it's able to do transcription and translation basically simultaneously. So it feeds the mRNA it's producing directly into a ribosome where it will immediately make protein. This does not happen in eukaryotic cells. They happen separately, one after the other. One, because of the nucleus, and two, because eukaryotic cells do this post-transcriptional modification that we talked about last video. The differential splicing of introns, exons, adding the cap, the poly A tail, that kind of thing. All of that needs to happen before translation happens, and so they're distinct and separate processes in eukaryotes, but in prokaryotes, they are happening simultaneously. So this just kind of shows an overview of it, of the central dogma. The image on the right is the more confusing version, but it has more info. But the basic gist is that DNA replication goes from DNA to DNA. That's done by DNA polymerase. Transcription is going from DNA to RNA. That's done by RNA polymerase. And then translation is going from RNA to protein, and that's done by ribosomes. So that's the general flow. We will be talking more about some specifics of how do our cells decide when to make these genes or not, which will kind of be the focus of the rest of this unit. But before we get there, we're going to talk a bit about these things called retroviruses. So, retroviruses, normal viruses, what they will do is they will inject, usually RNA, it could also be DNA, and then hijack the body's machinery to use their DNA or an RNA to make proteins, to make the viral proteins that then go on to infect other cells and reproduce the virus. Retroviruses are a bit weird, and they're more insidious, I think would be the best word for it. They actually inject RNA, but they also inject this enzyme called reverse transcriptase, which, as enzymes tend to do, the name tells you what its function is. It does transcription in reverse. So what reverse transcriptase does is it takes RNA and makes a copy of DNA from it. This DNA can then enter the nucleus and integrate into the genome of the host, whatever it is. So this is how HIV works, and is part of the reason why HIV is so hard to treat, because when the HIV virus infects your cells, it puts in the genetic information like all viruses do, but it also includes reverse transcriptase, and so it puts it into your genes. So the only way to get rid of it would be to somehow cut out the viral DNA from your own DNA, or just completely nuke every cell that was infected, which you can't really do when it's important cells like your white blood cells. That's your immune system. Don't need to know too much about viruses at all, really. They kind of tangentially come up in this unit with bacteriophages and now retroviruses. But they might ask questions about them, but they'll always give you context. But retroviruses, they go in reverse. They make DNA from RNA and incorporate that into your genome that then continues to reproduce. So that is it for this video. We are going to get into regulation of gene expression next. We're going to split that up a little bit, but this video is already a little too long for my liking, so that is all. See you later. I hope someone watches this before tomorrow. If you do, give me 2 thumbs up when you see me. I'll give you a piece of candy. Get out of here.